My job is to be the referee. So, 1, what are the 5 most critical questions? Go.
So the most critical question, and we've spoken a lot to this, and I'll be preaching Susan's gospel, but is, does early life exposure to the built environment actually influence health outcomes? I mean, we have evidence for yes, and we have evidence that it could be minor, and we have evidence left and right.
Minor in terms of taxonomy, though, could be major in terms of function. And I think we have to move past thinking in terms of taxonomy towards what are the actual mechanisms and functional outputs of these organisms in a distinct ecosystem, in the gut versus in the built environment.
Yeah. So just a question for you two is, why would you limit it to early life exposure and not make it exposure? Is it better to kind of break it up in hunks, or is there just already more evidence to suggest that it's early life exposure that matters?
I think at least the data from the asthma realm suggests that it's neonatal exposure, neonatal gut microbiome. The signatures that we see there, we don't see pan out at 12 months of age, and that's been shown in the child study. We have the same data in our birth cohort. So it suggests that I think that the susceptible periods are the times when the microbiome is developing in neonatancy and early life, and also I think in the elderly where the microbiome is disassembling, at least in the gut, that this may also be a prime time for susceptibility to colonization by built environment microbial exposures.
That doesn't mean that office workers, for example, in your particular interests, are not being influenced by microbial exposure in the built environment. It's just for us, that's a very interesting research area. So we could expand that. We could just drop early life exposure and go to just exposure. But under that, there are obviously these key areas. For behavioral as well as atopic diseases, there are very specific evidence to suggest that early life exposure is a critical element in the development of those conditions. But it doesn't start, we go perinatal. So pre, peri, and postnatal for the mother are also important. So it's super early life, if you will. And maybe even before somebody gets pregnant, you know? So early exposure in general could have devastating effects. So if but a lot of that comes down to lifestyle. But we just don't know what is the actual impact, and that brings it to epidemiological studies. More of those.
Okay, Joe, and then to the back.
So, can you hear me? No? Oh, okay. Yeah. Okay. So I totally agree with Jack and Susan about one's function, one's impact. I think in terms of maybe 5 questions, I think maybe another thing that is quantification. So people mention this. Most of the molecular studies is qualitative or relative. So really, you want to make a prediction in the future. So I think mentioned that you really need to know the biomass. So how to predict it.
Okay, do you want to turn that into a question?
Yeah, so whether the the question is that maybe say, can we predict, or then say, okay, whether the molecular biology to us is quantitative. How can we make this quantitative assessment?
That's a tool development question. That's what we need to do.
Yeah.
Is there a scientific question that we need to address with quantitative biology?
Yeah, scientific question I think more like retro prediction, right? Whether can we predict the future, the microbial dynamics, or the impact?
Okay.
So another question I think should be the mechanistic, the mechanism. What the mechanism controls the community dynamics?
Yeah. Okay. Dr. Gold's been trying to get recognized, and then we'll go to Dr. Haas.
I'd just like to append the first question or expand it to effects of the microbiome on susceptible life stages, because we've had mention of seniors, and also there are other portions of development, or if you want to call it decline, that may be susceptible life stages. Also, and this is just appending to the first question, whether reinforcement of exposure longitudinally influences the effects of the early exposure. Well, exacerbates or reinforces the protection, depending on which way the microbial community is working.
Yeah, I want to, I guess, ask a very overarching question, and that is, you know, given all the observations that have been made, are we necessarily looking at the right things? And let me back that up with an analogy. So, if I go out to the Potomac, I can measure, you know, gross organic measure of total dissolved organic. And then if I attempt to measure every single organic compound that's there, I'll probably only be able to explain these days maybe 60 or 70% of that total organic concentration by what I can measure. What is it that we don't measure with the microbiome, and could those non-measured things be responsible for some key adverse outcomes that we're really interested in? Or are we simply looking under the lamppost because that's where we have the light?
Could you condense that into a brief question? Yeah.
Well,
what do you say? What are the unknown unknowns?
No, it's not unknown unknowns. You know, what are the as yet I can't read what you've written there.
Looking at the right things, and the subtopic is what don't we measure that might have an impact.
That's good.
And I think what you were saying is that maybe we measure what we measure. If I understood correctly, you're also saying maybe we measure what we measure because that's what we can measure, as opposed to
Yeah. Well, yeah.
So
Jeff? Yeah.
the brief statement of the question is, can and should we intentionally manipulate the indoor microbiome? And two kind of sub points associated with that is, one, every time we do something, we have unintended consequences. So an example is we might introduce microorganisms into the indoor environment, for example, by having a dog, but that has other consequences as well that might affect other people in other buildings, might have other impacts. So there's a secondary consequence, and then there's also the issue of historically we don't do a particularly great job of maintaining complex things in buildings. So an example would be HVAC systems in buildings. I mean, we've had HVAC systems about as long as we've had buildings, heating and cooling and ventilation. And still, we're uncomfortable a lot of the time. If we were to look at the HVAC system in most buildings, it would be pretty disgusting inside from a lot of perspectives. And so now what we're talking about is introducing microorganisms, a very complex subject, and it doesn't stop with an introduction. There has to be a maintenance that historically we don't do a good job with.
Listen, one thing's weird though. You've got this company. I don't know if Jessica can speak to this. That company that is pumping the aerosolization of the microbiome. Brent showed it. Where's Brent? There he is. You showed it at the end, yeah? That little like probiotics for the home. Does that even have to have FDA approval?
I had the same question. I have no idea.
missed question two.
No, I know. That's my point is, that's if you're going to do
I'm going to ask, can I want to respond to Jeff's question?
Asked, and so I'm having trouble.
Can I add to what Jeff's question?
Okay, that's, yes.
So it's up there, could and should we manipulate the microbiology of the built environment? And my question for you, Jeff, is that we kind of are implicitly manipulating the built environment microbiome, or actually it's not implicit, we are. So how would you rephrase that in such a way that acknowledges that we are, but through our design choices, having an impact on the indoor microbiome?
So, I mean, I think that's very reasonable. We are definitely doing things that influence the indoor microbiome. But I think that there's a very big difference between saying, we're already doing something, so let's do something different, and implying that there's going to be positive outcomes because of it.
But are we at a binary of an engineered intervention? And I think with this question, and with all of these questions, we can adjust the level of resolution here. And I was wondering if in this dialogue, we're talking about the difference of implicit, yeah, we're here, where we have things on our clothes, or engineered interventions. We aerosolize something purposely to manipulate. So that's kind of what I was trying to do, is calibrate these two responses. And
again, it's probably implicit. Sorry. Again, it's probably implicit, but like adding the green indoor environment movement, you know, and there's a lot of companies out there, and you know, and again, that's one of those things that fails all the time. People put green walls in and let them die, but that's an explicit engineering.
yes, it's an engineered purposeful intervention.
But not for the microbiome necessarily, but for sometimes microbially mediated metabolic products and chemical transformations. There's a geomicrobiologist who's working on a paper at the moment. It's very good. It deals with all of that. Can't talk about it.
Okay.
Are you still the same question?
Oh,
or
yes,
no
same
one?
question, yeah.
Same.
Okay, so yeah, I think so before we talk about engineering the communities, maybe we really need to answer the questions, why the community is stable? If the community is not stable, if change like a few minutes, like within one day, so you don't know how to engineering. If you engineering, you cannot sustain the community if not stable. So maybe stability is the critical questions.
If I could take a stab at turning that into a question, we seem to be having a lot of statements and opinions and amendments and fewer questions. But are you asking a question about what is the dynamism of this microbial community? Or
Yeah, you can answer.
What are the
So why is that?
temporal dynamics of?
Yes.
Yeah, something like
So
that.
why is the microbial community in the built environment stable? So
Or it's more than is it stable, because we've already said it's not stable, right?
It is stable if you have stable occupancy. I mean, all the environments we've looked at over time have been stable with stable occupancy and stable conditions. You know, even the hospital, that was 365 consecutive days, and it really didn't change very much. It only changed when the occupancy of the rooms changed.
But to counter that, also, you have to be careful. Surfaces versus indoor air.
Oh, yeah.
Well, what,
So just to reiterate, so this... no time. Sorry. When you look at surfaces, what I think are impacted very differently from indoor air concentrations and communities are going to change on a daily basis. Andy will talk about this tomorrow. On an hourly basis, potentially based on driving forces of infiltration and ventilation rates and so forth. So we have this very big distinction between who's studying the indoor air and who's studying indoor surfaces.
and maybe that,
Oh, I was just saying that I was just helping out with this question that I think going back to asking whether or not the indoor microbiome is stable is a relevant question, because there have been some studies showing that it's stable, but in the grand scheme of things, there haven't been a huge number of studies in all different types of built environments across all different media in the indoor environment.
Yeah, okay. Okay, that's good.
Yeah, I'm going to add one, although what I heard from this conversation was what building or what built conditions establish stable microbial environments. That's what I heard. So if we wanted stability, what would we do? And if we are okay with instability, what is that? Okay, I have a slightly different one, which has to do with the issue of air and surfaces. So if I understand, given the impact of human systems, which from what today's conversation was, were 3, respiratory, digestive, and skin. I heard 3, maybe there's more, but I heard 3. Which built environment transport mechanism, is it the air, the water, surfaces, human to human, or the outdoors, is most critical to human health? That's my question.
Very well stated.
I had plenty of time to write it down. I wrote it down. Okay. The impacted human systems of respiratory, digestive, and skin responses. That's my assumption, is where we're starting.
Which transport mechanism?
Which built environment transport mechanism? And then I have a list, but I don't know if it's complete. The air, the water, surfaces, human to human, and the outdoors, which sort of will translate into air and water and other things, I don't know, but or animals, is most critical to human health.
What mixture of?
Well, human to human, right? Human to mammal?
Do I get to add a question? Yeah, how does human behavior affect the microbiomes of the built environment? We've been, I think, leaving out this aspect of what the people are doing in the built environment. So we want something about not just the static stuff that scientists measure, but the kinds of things that humans do.
Where is the engineer who set the thermostat for this room?
Exactly. Good example. Yes.
Yeah, I'm getting a real sense of, the chicken and the egg here, in that we're interested in this because of how the built environment affects humans. But we've also found that humans are the greatest impact factor on the microbes, and so which comes first? Are you going to have to change humans in order to change the built environment? It would seem to be a logical conclusion there. The other thing is I look at the examples of fecal microbial transplants in the gut, and you see if you just put in organisms, very few of them take. The whole thing is pretty stable, unless you condition the patient with antibiotics to, in essence, clear the ground. What I'd like to see is an experiment in the built environment where you use something like aerosolized hydrogen peroxide to completely, pretty much sterilize a room, get into nooks and crannies and things, or a hospital or whatever, and see what repopulates and how it repopulates. And is it different?
We've done that in restrooms and in hospital rooms, and it was... We've done that in restrooms and hospital rooms, and yes, you do see very specific succession over an hour-to-hour period up until it reaches a stable community. In restrooms, we repeated that in 4 different restrooms in the same building. Total sterilization, then monitoring it every hour for 8 hours, and then every day for 6 weeks. and you see within 5 or 6 hours, the community stabilizes. And pretty much, you could tell girl rooms from boy rooms, right? And you could tell high use from low use. But on the whole, there were stable characteristics in the community structure. And in the hospital, the same thing happened. So the rooms were comprehensively fumigated and sterilized with bleach, 10% bleach on surfaces to burn off all the material, and then they populated.
You have a centralized air handling system.
Well, this is HEPA filtered, right? Or would we consider that as a major source, Brent? For the hospital?
Thanks. And I want to point out that we're having a dance between what was rather artificially in this outline. You know, the notion was we were going to ask the big meta-scientific questions and then follow up with suggestions about how to answer them. And that's a good example of a way to answer a question, experiments in which we wipe stuff out and see what comes back. And so it's good that we're getting these questions down, but when we come up with a new outline, I would say that we may reorganize some of the good questions that have been thrown out. Yeah, Chuck again.
Okay. So what's the role of mixed exposures in relation to these microbiome, either whether it's adverse or good effects on humans? And I just wanted to make a comment to this fact that talk about stability. So Jack was saying that the rooms become stable over time in the hospital, but if there'd been an upset condition such that the walls became wet from a leak, and you'd have a complete change so that the humans weren't the things driving it, I'd like to know what happens and how it upsets the conditions. I can't shorten it. And the one thing I was going to say, you wanted 5 questions, but I know, like in brainstorming, it's good to get like 100. And
I agree. We're going to get a lot of them down. I agree. Yes. Gary.
I want to ask a very basic statistical question. And this is going to expose my naivety for the microbiome people. Are we measuring something that's real in the sense of what is the intrinsic reproducibility, even on split samples, of the microbiome relative composition information that's being measured and reported? Unless we know that, we can't understand are differences between sites, treatments, et cetera, real or not.
So,
just as an extension to the comments I gave during my talk, you know, I think just understanding how the microbiome might shape significant health disparities that we know of. I mean, of course, that's a big question, and my guess is it's well beyond the built environment, and we have some suggestion on how that might be the case for allergic disease, but I think it should be a focus embedded in some of the questions we're asking.
Jessica.
So I have 2 questions that are related to all of the other questions, but I think it would be useful for us to explicitly consider how building and city design in response to global change, so climate change and the rise of megacities, how our increased population sizes, how both of those future outcomes have the potential to influence the indoor microbiome and human health. And the second idea I had is understanding how landscape architecture relates to the indoor microbiome, so the abundance and diversity of plants in the outdoor environment.
Nice. Ben. So.
Yeah, because you need to answer. Human microbiome versus the microbiome. The parsing of those 2 words, I'm trying to understand.
The difference between human microbiome and just microbiome? Yeah. A microbiome is the collection of microorganisms in any environment. So you can have the marine microbiome, you can have a human.
This is microbiome in the built environment.
this, yes.
And then you were talking about the human, and the human microbiome.
No, I'm talking about the microbiome of the built environment in response to design changes, the choices that architects and engineers make to deal with global climate change, for example, or increased population sizes. How will that influence the indoor microbiome, and does that matter to human health?
Okay, Lynn.
Just one of the things that strikes me is that we're very kind of unidirectional in our consideration of the built environment influencing the microbiome, and how does the microbiome actually influence the built environment is another key question. Does the types of organisms that are there influence the exposome in the home, and how does that happen?
And I'd like to make a commentary, which your remark reminds me that in this entire discussion we have been, making the assumption that we are living in these built environments, and we know a lot about what happens to built environments when we move out. They biodeteriorate, and we haven't spent much time looking at, what happens to abandoned homes, and it's amazing how rapidly nature takes over and the building components get destroyed. So I think that we haven't talked today about this need for a glossary and definitions, but I think before the next meeting, the staff and I will give more thought to that. We've distributed one that's been developed, and we don't necessarily want to be reinventing the wheel, but I've noticed today that by not having reviewed some ground rules of stipulated definitions, that we may be talking at several different levels. But okay, go ahead. Yeah. Mark, do you?
Is this on? You
I
hear
don't
me
know.
okay? Yeah. My question is, and I'm going to borrow Susan's term of exposome. I like that. So how do cleaning practices, what's the diversity of cleaning practices, and how do they affect the exposome? I'm trying to make that big question. And can we extend that diversity of cleaning practices to those that are, considered common routine, soaps, cleaners, and so on, and more of a physical plant and infrastructure cleaning practice, like Jeff was relating to HVAC systems being, rarely cleaned. They're maintained, but perhaps not cleaned. So
Could you take a stab at defining exposome, if we're going to make this a question?
Are you asking me?
Yeah.
Okay, so I'm going to come back to what was just elegantly described as what do we touch, breathe, drink, and those pathways: surfaces, aerosol, water, and what was the fourth one, Vivian? Food. Well, okay, I guess food, but skin. I'm just going to keep, I won't be so elegant, I'll just be gross. Surface, breathe, drink. Okay, so those are it. But I want to get to the diversity of cleaning practices. No, I don't want to be that fine yet. I'm trying to just kind of stay out of bigger picture of resolution. And of course, there's chemistry. Some are surfactants, some are oxidants, some are, but can we box those up and get some systematic way to see how those might affect how we're exposed to the diversity of microbes in the built environment once we agree on how to measure that? Different. Anyway, that was my call on it.
I may not be stating this correctly, but how do the indirect effects of the built environment microbiome, which could either be the dust, the air, or water, influence health? And what I mean by indirect would be breakdown of chemicals in the household and therefore creating different chemicals, or breakdown of the surface layer of the pipes. And then just one comment that perhaps the reason why we focused on certain health effects like asthma and respiratory infection and skin disease is because we haven't really been focusing much on the microbiome of the water this session, whereas we were focusing on it last session a little bit more.
Mm-hmm. Might this be a good time to talk a little bit about methods? Or no, I would like to hear from the associate.
I just wanted to ask one question. It's not a research question, but I don't. Is anybody doing any kind of research that kind of a impact profile when you bring all of these exposures together across all of the different risks that we've talked about? Is there any such project underway?
Not to my knowledge, but perhaps someone else in the room knows. Chuck?
Well, I mean, there are methodological approaches for integrating multiple adverse effects. The use of qualities and dollies, for example, is a metric to integrate diverse types of health outcomes into a single unit. But then you have to integrate that by estimating each one of the inputs into the dolly and quality equation.
So I'm thinking of, for example, we've talked about people who live in disadvantaged communities, and so if to take a snapshot of where they live and then actually piece out all the competing risks that have to do with the kind of research we're talking about here. Is that, I mean, it's got to be possible.
Oh, no, it is possible, and actually it's an interesting question, and you know, maybe some of the folks from public health schools know more about the detailed methodology, but using dollies and qualities, in fact, you can factor in access to health care as well, which was part of the disparity issue in terms of outcome weights. So methodologically it's possible, and there are metrics to do it. I don't know that anybody has done it in this context.
Yeah.
So on that subject, one of the biggest frustrations for me over the last few years is the realization that no one's ever done a real burden of disease quantification. I mean, someone can correct me if I'm wrong, but of housing conditions and health in the United States, which is very much connected to like the fact that the average family we work with doesn't have one problem. They have 25 problems, and they might have 12 housing problems. And so putting all that into context and quantifying the health burden, quantifying the cost to society is something, there's been a little bit of this work done in Europe, but to my knowledge, we've never really done that in the United States.
We only have a limited amount of time today, so perhaps this would be a good moment, even though we haven't centered around the questions, to talk a bit more about methods and ideas that people have for places that we need to develop better methods. We were kind of just talking about this, you know, integrating several studies and coming up with some generalizable conclusions, but I think there are a lot of other methodological issues. How? And then. Last 135, based on Brent's citation of that paper.
I see a lot of discussion that, or presentation of studies that in some sense are replicating a portion of studies that have been done in the past on particulate matter. I mean, you know, microbes are particles, and it's interesting. I had occasion to contact two of the pioneers of the indoor air research field Charlie Weschler, who you know, Ruth, and Bill Nazaroff, and I wanted to ask them for their first indoor air publication. And in both cases, I had in my own mind misidentified them, but they each studied the effect of air quality on objects, not on people. In Charlie's case, he was interested in switches in telephone company switching offices, which is what his employer paid him to look at. And in Bill's case, he was a graduate assistant researcher at LBL, and radon was the pollutant du jour when he was doing that, and so he studied radon. He went on to do his PhD research on the effect of air quality on art objects.
Yeah.
And it just strikes me, both of those two, pioneers did that work more than 36 years ago. And so much repetition in terms of methodology with this research on the indoor microbiome that we at least need to mine what's known.
Yes.
From the work that's been done about methods for non-microbial indoor air pollution.
That's an excellent point. It's an excellent point. Yes. Brent.
So I have two thoughts on methods and instrumentation and tools. One is kind of the obvious of quantification that just isn't done in a lot of studies. So these are, I don't know if the right thing is to have some panel array of instrumentation or of analytical techniques that all studies include, right? That include culture-based, that include relative abundance, that include metagenomics and their outputs, include live dead viability and so forth. So there's some sort of parallel when you think about outdoor air pollutant monitoring stations, right? You've got to get whatever monitor you've developed and decide to use. It's got to be registered. It's got to pass this federal, registry to be an equivalence method. So I don't mean to advocate for every study should use this same kind of instrumentation, but there is something that we might be able to learn there. And whatever you can get towards quantity and live dead and more sophisticated measures of what they're doing, for lack of a better word. And then sort of beyond that, most of these are time integrated, somewhat long-term samples with low biomass detection limits. So the pie in the sky is real-time instrumentation or real close to it that can give you real-time analytical methods or real-time analytical results. Did you get most of that?
Yeah.
He was teaching the non-scientific science. Can you say the second half again? I got to the live and dead.
Yeah. Oh, okay. So you got the kind of combine the tools that are out there now in more studies. Yeah. And then the next one was most of the samples that we've talked about today are time integrated. So whatever you can do to move towards higher time resolution measures. Mm-hmm. So this would be supporting instrumentation development and that sort of thing. Basically, we've got fluorescence instruments here. Mark's done some of this for bioaerosol fluorescence, that kind of thing. So there's just not much out there.
That's true. Jake, you had your, yes? Yes?
Yeah, he took it.
Oh, okay. Okay. Okay, sorry.
Am I on now?
Here, we'll bring you this one.
Give me a mic that works. I'm going to speak.
There's one that doesn't work. Let's put it away.
Okay. So this is the non-scientist perspective. It's more the engineering perspective, which is all I have to offer. So here's my question. Is the dry building microbiome more or less desirable than the damp building microbiome. And if it's more desirable, if the dry building is more desirable, then I believe more study is needed. And if the wet building microbiome is more desirable, then I believe more engineering is needed. Because then it's just a matter of managing moisture, isn't it? But if it's better to if it's better to be wet, which I don't believe, of course, but if it's better to be wet, then we got to figure out how to make things wet. That's all.
Oh, down now. Okay, did Hal, oh, sorry.
Just on the topic of methodological data gathering, it seems to me that, it's been mentioned that there's a kind of a stronger need for functional characterization and functional assays. So I think one hypothesis is, can we start to develop more high throughput, lower cost functional assays that are sort of comparable to what's being done right now with 16S. So I'm just thinking for myself personally, targeted sequencing, that would be somewhat, still targeted, but somewhere it's in between sort of untargeted metatranscriptomics, but would be more sort of quantifiable targeted gene expression in a microbial community or something like that.
Okay, we need to wrap up pretty soon, and so even though we could stay on this for a lot longer, and we will be returning to it, I want to raise this third question on our written assignment here about the who. And we're very science, building science, architecture, engineering representative in this study and in the speakers we've heard. Do you think that we need others to join this conversation, and who are they? And let's talk a little bit about the experts. Andy, you haven't had a voice yet.
Yeah, when you said experts, it kind of threw me off. But I mean, if we're talking about impacting design and maintenance, we need to talk to the people who design and maintain buildings. Okay, there's designers who are, engineers like Dennis, and then there's designers who aren't like Dennis. But then the people who maintain the build, who own the buildings, who make the decisions about, how much they're going to spend on the design, and which of the design recommendations they're going to toss based on the budget, and then, the people who maintain the buildings, both who hire and direct the people who clean or don't change filters, and then the people, do they still wear blue shirts? Who work upstairs or downstairs to do all that stuff. So there are professional, there are trade associations of those folks that we might want to talk to, though sometimes some people listen better than others, that's all.
So sort of taking up my previous point about learning from the last 40 years, what we saw in indoor air quality was at first there was a small number of experts that became the consultants du jour. And then as specific problems emerged like asbestos abatement, lead abatement, mold abatement, so on, different industries and trades that had the equipment to do those things and had access became lead, mold, and asbestos abatement contractors. The asbestos guys, they pretty much put themselves out of business because they removed it, but then came mold, and they love mold because you remove it and it comes back.
I see.
But so then after a while, the the people who used to maintain your household level or small building HVAC equipment and systems, contractors, all of a sudden they were also IAQ specialists, so and duct cleaners. So you, as the public demand for the services expanded, the kinds of professionals that got involved expanded, and it's really looking at it from the other side is who is a homeowner or a building owner going to call when they think they have a mold problem or a microbial problem or Legionnaire's disease or whatever it is that inspires them to go on the web to search for somebody to provide services to help them. I was going to say look in the yellow pages, but, well, you all know I'm not young, so.
Any other input? Yes, Jessica.
I've just found from my experience at the University of Oregon that speaking or working with landscape architects on this has been valuable only because they are investigating what's happening with the outdoor microbiome.
I just realized that my earlier comment reflected a commercial building bias, and so shame on me. You know, if we're talking about design and maintenance and we go to the residential side, it's a whole different picture on maintenance. You know, I don't think there's a, you know, American Society of Homeowners, but, you know, somehow if we're trying to send a message to the people who maintain their own homes, we need to engage, you know, with somebody.
So there again, we can learn from radon, and it was the real estate industry and the National Association of Home Builders that got involved early on in addressing the problem, and the American Lung Association, who was willing to take EPA's money and distribute it.
And if I could interject, Hal just gave us, I think, an important clue that one group of individuals that we certainly could be engaging are professional societies of these various expert groups that have been studying it from different directions. For example, lungs, heart association, microbiologists. I believe Kathleen had her hand up, and she also hasn't had a chance to talk before. So let's give. Oh, I'm sorry. You have, and then we'll go to Kathleen.
Okay. So I wanted to draw attention actually to those. Gary, you guys handed out, when you worked with those green homes, you handed out those booklets, right? And part of those, so I'm thinking of housing and urban development being a conduit for getting information out. So you could probably describe, can I hand this to you and have you describe those booklets and what information was in them?
Sure. So this was normally I have a copy with me. I carry it everywhere. But these were booklets that we developed. It was actually, the development of the book was paid for by our HUD grant, and they were meant to be educational materials handed to every resident who moved into a green unit in Boston. And it was everything from how to keep your new home healthy to how to save electricity. These were residents who had never paid for utilities prior and now get utility allowances. It had everything from how to use a thermostat, because people had never had thermostats, to how to keep your home healthy and things like that. I'm happy if anybody's interested. It was, you know, funded by our grant, so you bought it. It's paid for by HUD, and so the Boston Housing Authority, every family who moved into a green unit got one. They were in English and Spanish. BHA paid for translation into Chinese But once again, it's, for those of you familiar with like the 7 principles of healthy homes, that's very much embedded in there, but it contains other elements.
And so I let you go ahead and ask your question because I thought it would probably lead to my comments. I had asked early on what the purpose of this was, and the answer that I got was that we're going to write a report, and the report is going to help us understand how agencies should direct their funding for research. But what I'm hearing in the room is that because the microbiome of the built environment has a health impact, we're talking a lot about communicating to people who are not researchers. We're talking about communicating to professional societies. We're talking about communicating to people who own homes. And if we're going to do that, I would actually recommend that we go right back to Leanne's talk and think about including some marketing help. And I don't mean marketing in terms of how to sell things, but in terms of the appropriate communication of the technical information to folks who don't have a technical background, because I hear a lot of discussion about communicating to those folks as well.
Thank you. Chuck?
One other sector that has a lot of skin in the game is the insurance industry.
Thank you. And something I was hoping someone else would raise, and since I haven't heard it and we're running out of today's time, I'm going to throw it out there, is that, you know, we haven't been doing a very good job of asking people, you know, quote, the ordinary people, what worries them, you know, the mothers of small children with asthma, or people who are in rental places where suddenly they feel like mold or something. And we often ask our questions over here and don't do a good job of connecting them to the other. And the guy who was talking about the ants, you know, you give a lecture on the ecology of tropical ants, and then somebody says, how can I keep the ants out of my kitchen? And I think all of us have been faced with some of that in our research, that you give your lecture and then comes back what you might think of as a simple question that has very little to do with it, but there is a disconnect. And so if we're going to communicate our findings, we really need to do a better job of asking people who are living in houses who are not thinking about all this all the time what their worries are. Yeah. It's 5:30. We have dinner reservations at 6. It's on your schedule. I can't remember the name of the restaurant, but apparently it's about 3 blocks from here, and the staff is talking about maybe having a caravan that walks that way. I think it's appropriate that we start to wrap up because I know my brain is full. And if we have 1 or 2 last burning remarks, otherwise I might move that we decompress and start thinking about dinner.
I don't know if this is burning, but I'll forget it if I don't say it. And this is a little bit about the WHO, and there's an earlier comment, you know, question whether we should regulate or not. We regulate right now. We have building codes.
Yes.
And they tell you, you know, let's not talk about compliance or any enforcement or anything like that. They tell you how much outdoor air you need to bring into a building, how crawl spaces are vented, how attics are vented if they're vented, and, you know, a whole host of other things. So, okay. Anyway, so there are some regulations, there are building codes. Maybe this is something to talk about,
yeah,
covering in a future meeting.
It's a good idea.
I didn't want to forget. You can decide whether it was burning or not.
Okay. Plans for tomorrow. We're going to have another day here. with starting around 8:30, and the first thing on our session is reports from breakouts. Since we haven't had breakouts, I think Katie and I are going to have to do some discussion about how we're going to rearrange that particular aspect of our agenda. But we will come back tomorrow with a more specific game plan, and I want to thank you all for sticking with us and having so many good ideas and being such a wonderful study group and participant group. So I hereby adjourn us for day one.